Welcome everyone. Very nice to meet you all. Thanks for joining this talk. I'll just, wanted to give a brief overview of my research, what I've done in the last few years. So I'm currently a university teacher in marine science at the University of Edinburgh. I joined Edinburgh about like 2.5 years ago as a postdoc, right after I finished my PhD at the Alfred Wegener Institute in Germany. My research mainly focuses on the effect of climate change on cold water corals. So I've done several aquarium experiments of different durations, most of them with multiple environmental drivers over the past few years. So just wanted to give you a brief overview over my research, and I'll focus on the two largest experiments that I've done so far. So I'm mainly interested in studying the physiological response of cold water corals to environmental changes. And I'm also interested in looking into like how the environment affect their skeleton, how ocean acidification affect their skeletal dissolution, and so on. Just a quick introduction to cold water corals for those of you who might be less familiar with what cold water corals are. They're globally distributed, as you can see on the map here. No locations are shown in blue. But we can mainly find them in cold and deep waters. So they mainly occur at like a few hundred or few thousand meters depth. And there are only a few locations around the globe where you find them in shallower waters. And of course, because they occur mainly in the deep sea, there's no light. They don't have any symbiotic algae, so they completely rely on heterotrophic feeding and collecting like zooplankton from the water column, their food source. There are different kind of species. Some of them are solitary, but some of them can form large colonies, just like tropical corals do. So they can form really complex three-dimensional reefs, which are associated with quite high biodiversity. So they're important ecosystems and habitats for other marine species, benthic species, but also fish species. But of course, they're also affected by environmental changes and climate change. So here I've shown a figure from Andrew Sweetman's paper that summarizes the main environmental changes at the deep sea, at the sea floor. So these corals will be affected by a combination of warming, ocean acidification, changes in oxygen concentration, so deoxygenation, and changes in food availability. So we expect that the flux of particulate organic carbon to the seafloor will decrease in the next few decades due to climate change. So what we know so far is that cold water corals or live corals can actually grow and survive at low pH conditions. They might not be affected by ocean acidification too much. We find them at some locations at aragonite undersaturation. So this means the corals, they build their calcium carbonate skeleton out of aragonite, and once the aragonite saturation is below 0, this skeleton will start to dissolve. Nowadays most corals are located at aragonite saturation over 1, until the end of the century, it's projected that about 70% of cold water coral habitats will be subject to aragonite undersaturation. So I'll go a bit more into detail this later on in the talk, but that means that the bare coral skeletons will dissolve. However, the live coral, they are still covered with live tissue, will most likely not be too much affected by ocean acidification in the future. We know from like several aquarium experiments and field experiments the elevated temperature can affect their physiology, but this depends on the species, the location, and the extent of warming, of course. And there's very little research on the effect of changes in oxygen concentration on cold water corals. There are only very few studies that have looked into this, and they found quite contradictory results so far. So we don't really know how like reduced oxygen concentration will affect cold water corals in the future. And the same with feeding. So we expect that increased feeding may help them to compensate for like negative effects of changes in other environmental factors. But there are very few studies that took into account the changes in food availability. And I've said before, we actually expect a decrease in food availability due to climate change because of changes in primary production and decreases in primary production. So what was the motivation for my research? So mainly that there's little information on the combined effects of multiple environmental drivers. So far, most experiments have looked at the effect of either ocean acidification or warming on cold water corals, or combination of these two. I haven't really taken into account any other environmental factors. There are very few long-term experiments. So most experiments so far have been run over a few weeks or months, but really only a few that have run for longer than half a year or even a year. And then, as I've said before, there's very little research on the effect of changes in food availability or oxygen concentration on cold water corals so far. And as I've also mentioned before, we do know that ocean acidification will affect the dead skeletons of cold water corals, will lead to skeletal dissolution. But we don't know yet over which timescales this will happen. As I've mentioned before, these cold water corals can create quite large three-dimensional reefs and three-dimensional frameworks. And it's important to note that a large part of these reefs is actually not covered with live corals anymore. So a large part of these reefs consist of only dead skeletons that might be affected by ocean acidification in the future, which can then have an effect on the reef structure and integrity. But we don't know yet over which timescales this will happen. So I'm going to start with the first long-term experiment that I've done at the Alfred Wegener Institute when I was doing my PhD there. And in this experiment, I looked at the combined effect of changes in temperature, pH, and food availability on different life stages of the solitary cold water coral, Coryphylla huinayensis. We collected these corals in a fjord in Chile, and this research was done during COVID, so it's actually changed quite a lot because we weren't able to go on expedition to Chile as we had planned, and we weren't able to do like the experiment that we had planned to do. We were planning on working with a different species, Desmophyllum dianthus. But yeah, we weren't able to get any new corals, but we were lucky enough to have a lot of Coryphylla huinayensis in our aquarium tanks at RV. And they were actually reproducing in the tanks, so we had quite a lot of corals available. And I thought it would be really interesting to not only look at the effect on adult corals, as most studies so far do, but also take into account like potential effects on different life stages. So that's what I've done for this experiment. So yeah, this experiment was run for 6 months in total, and I looked at the single and interactive effects of changes in aragonite saturation, temperature, and food availability. So I had 4 different treatments, one like ambient or control treatment, one aragonite undersaturation treatment, elevated temperature treatment, and then a combination of elevated temperature and reduced pH or reduced aragonite saturation. And for all of these 4 treatments, I had a high feeding and a low feeding treatment as well, as you can see here. And we've included 3 different life stages. We looked into the effects on early juveniles, late juveniles, and adult corals. And first of all, we had a look at the health status of the corals. So depending on how, on the tissue covered surface area and how much of the skeleton was covered by tissue during the experiment, we put them into like different health categories. So you can see here on the left, it's a coral polyp that is completely covered with tissue, looks quite healthy, whereas on the right side, it's obviously dead because there's no tissue anymore, and you can just see the bare white skeleton that's left. So we checked the health status a few times over the 6 months of the experiment, And as you can see, yeah, we don't really see any differences in the first 3 months. So you always see the 4 different treatments at the top, the 3 life stages at the bottom, on the left, the early juveniles, and the right side, the adult corals, and then the low feeding treatment at top, high feeding treatment at the bottom. And yeah, you can clearly see we only saw clear differences between the treatments after 6 months of the experiment, where the corals in the elevated temperature treatment and the combined treatment of elevated temperature and reduced aragonite saturation state were clearly negatively affected by these treatments. And quite a lot of these corals died over 6 months. This graph showed the change in tissue cover of the different life stages and the different treatments. And here you can clearly see that the early life stages, so the early juveniles and the late juveniles, were doing quite well in the ambient and aragonite unsaturated treatment. They managed to extend their tissue coverage in the high feeding treatments by more than 50%. But we see a clear feeding effect here because they have a much lower tissue coverage in the low feeding treatment. So they weren't able to extend the tissue coverage and the low feeding treatments. And they will also reduce their tissue coverage in the 2 treatments with elevated temperature. So you can clearly see a decrease in the tissue cover in these 2 treatments. And we see a completely different picture for the adults because, they reduced their tissue cover in all 4 treatments and under both feeding conditions. So yeah, a clear difference between the life stages and the treatments here. This graph here showed the calcification rate of the 3 different life stages and the 4 different treatments. This first plot here only showed the results for the high feeding treatment. Here you can see that over time, the early juveniles and the late juveniles increased their growth rates in the ambient and aragonite undersaturated treatments, where they decreased their growth rates in the temperature and combined treatment. And it's quite interesting to note here that they were able to increase their growth rates even in the even at aragonite undersaturated conditions, which might be contradictory what we would expect. But yeah, it's important to highlight, as we've seen in the plot before, that the corals were still completely covered with tissue in this case. So they weren't really affected by aragonite undersaturated conditions because their skeleton was protected from the surrounding seawater by the live tissue. So we didn't see any skeletal dissolution in this case. And then, important to know that in general, the growth rates of the adult corals were quite low, which is also something we would expect. That's usually the case for cold water corals. And then when we look at the effect of the low feeding treatments, you can clearly see that growth rates decreased in all life stages and all treatments. So the corals definitely did not receive enough energy to maintain their growth rates in the low feeding treatment. On this slide here, I've summarized the results that I've shown you before. So this first part of the graph only showed the results for the ambient treatment and at aragonite undersaturation. And I've plotted the effect size of the somatic growth and calcification rates here for the 3 different life stages and the 2 different feeding treatments. And you also see the survival rate in percent next to it. So here you can clearly see that the early life stages received enough energy to maintain and even increase their growth rates. So their energy demand was definitely met. They were able to grow faster, at least in the high feeding treatments under these treatment conditions. But we see a clear feeding effect because, the growth rates were a lot lower in the low feeding treatment. And then for the adult corals, they clearly did not receive enough energy in any of these treatments. We didn't see a difference in the feeding regimes. So the energy input was not enough to really meet the energy demands in this case. If we have a look at the 2 other treatments, elevated temperature and combination of elevated temperature and aragonite undersaturation, you can clearly see that none of the life stages received enough energy to meet their energetic demands. So the growth rates decreased in all of these treatments, and as you can also see, quite a decrease in survival rate, especially in the adult corals. So yeah, just wanted to highlight with this experiment how important I think it is to take into account the effect on different life stages, not only focus on adult organisms, but there can be a clear difference between life stages, and that's of course important to take into account when we want to study the effect of climate change on like a whole population. We need to take into account like recruitment and reproduction and so on as well. I'll now move on to the second experiment that I wanted to talk about. This is one that I did at the University of Edinburgh in collaboration with the Marine Station in St. Abbs. And here I looked into the combined effect of changes in temperature, pH, oxygen concentration, and food availability on Desmophyllum pertusum or Lophelia pertusa. And in this experiment, we looked at the effect on live corals, but also on the effect of ocean acidification on dead coral skeletons. And this experiment was run for a whole year. We had 7 different treatments, so we weren't able to look into the combination of all of these factors because it was a bit too complicated to do that. But we had a control treatment and then 2 multiple driver treatments, one with high food availability, one with low food availability, and a combination of reduced pH, elevated temperature, and reduced oxygen concentration. Then we had 1 treatment with only reduced oxygen concentration. As I've said before, there have not been that many studies on the effect of changes in oxygen concentration on cold water corals so far. And in all of these corals, we looked at the effect on live corals. But then we also had 3 different pH treatments where we only looked at the effect on ocean acidification on dead coral skeletons. So this first graph here shows you the survival rate over the whole duration of the experiment. And as you can see, the survival rate doesn't really differ that much between the different treatments. It's a little bit lower in the 2 multiple driver treatments, but really doesn't differ that much. But then when we look into the growth rates of the live corals, you can see a clear difference between the 2 multiple driver treatments and the control and reduced oxygen treatment. So after more than 3 months, we can clearly see that the growth rates in the 2 multiple driver treatments decrease quite a lot. We've only measured negative growth rates in these 2 treatments for the second half of the experiment. And this is most likely due to the skeletal dissolution in these 2 treatments with low pH conditions and occasionally aragonite undersaturation. And quite important to note that in this case, our fragments weren't completely covered with tissue. So this is different from the previous experiment because there were larger fragments with like multiple polyps, not the whole fragment was covered with tissue. This is why we did see, did find these results of negative growth rates because of the dissolution of the bare skeleton. And the same as with the previous experiment, I think it's again important to note that we found this delayed effect. That it really takes like 3-4 months until you actually see like differences between the treatments, which is one of the reasons why I think it is so important to run like long-term experiments, because you might find like very different results if you only look at the effect over a few months rather than a whole year in this case. I also measured the energetic reserves of the live coral. So I measured the protein concentration, carbohydrate concentration, and lipid concentration of the live corals. This is just a combination of all of them to show you the total energetic reserves. We did not see any significant differences here, as you can see, but I kind of expected to see this trend of like lower energetic reserves in the 2 multiple driver treatments, especially the 1 low feeding treatment than in the control and oxygen reduced oxygen treatment. And now moving on to the results of the dead skeletons. So this first graph here shows you the weight change of the dead skeletons and all of the pH treatments. And as expected, you can see that the skeletal dissolution rate was highest at lowest seawater pH. So when the aragonite saturation decreases, the coral skeletons start to dissolve as we would have expected. And here you see the results for the control and one of the multiple driver treatments over time. Again, just wanted to highlight, kind of the delayed response. As you can see, after 3 months, we didn't see a difference in the skeletal dissolution between these two treatments, but they only became apparent after more than 6 months. And then you can see at the end of the experiment, we've lost about like 40% of the skeletal volume of the corals in the multiple driver treatment. This is just a visualization, just an example of 2 samples from these 2 treatments, just to give you an idea. So we've scanned both all the samples, all the dead coral samples with micro CT. And yeah, here you just see some images over the time of the experiment. And you can clearly see in the low pH treatment in red, you can clearly see the skeletal loss at the septa, but also like the outer rim of the skeleton, where the control sample still looks exactly the same as at the beginning of the experiment. And these 2 videos are not from my experiment, but a previous one that Sepp Hanniger did. But again, just a visualization of what we see. So on the left side, you see a live coral sample that was still covered with tissue. On the right side, you see a dead coral skeleton that was not covered with live tissue anymore. And both of them were collected at a location with aragonite undersaturation. But you can clearly see that only the sample on the right side was affected by acidified seawater, where the sample on the left side was protected from ocean acidification because of the live tissue cover. And we want to do like similar analysis with the samples from my experiment. It's just like a really large data set, so we're still in the process of analyzing all of them. And yeah, this is kind of the same, like a video of the micro CT images of 2 samples, a live coral and a dead coral, and you can clearly see the porosity in the skeleton on the right side over time. But yeah, that was all from my side. Thank you very much for listening. Thanks for your attention. And I just wanted to highlight that with these experiments, they're like quite complex, and running experiments over such a long time and with so many environmental drivers, it's quite complicated. So I had a lot of help from a lot of other people. So just wanted to mention them here and thank them. Lots of students have been involved in these experiments, lots of technicians, the staff at the marine station in St. Abbs, and so on. So yeah, I had a huge team to help me with this. Thanks a lot for your attention, and I'm happy to take any questions.